There we go. Perfect. Thank you. Over to you, Andrew.
See the right slide there.
It's perfect. Thank you.
Right. Okay, so let me see if I can get this to work properly. This is the first one I've done as a talk on Zoom. So, well, right. So, thank you for organising this, Rob and Joe. And I think, as Rob mentioned, one of the things I think we're quite keen to get an idea of is the range of different questions and approaches being used with ex vivo or in vitro culture models to mimic aspects of the tissue microenvironment, and how we might be able to relate that then to early detection approaches. So one of the things I'm principally interested in is how the mechanical properties of the extracellular matrix microenvironment influence cell behavior, and how that affects how mammary epithelial cells in particular might be predisposed to becoming oncogenic. So in that context, there are a number of things that we're trying to do in the lab to develop ex vivo models to recapitulate aspects of tissue function. And some of the things we're doing are to look at the primary tissue of high-risk women, their breast epithelium, and what the characteristics of the tissue are, using approaches like laser microdissection, mass spectrometry, mechanical measurements like AFM and rheology, using then approaches where we can mimic those tissue features in a model, and then use those models to look at functional characterization of what's actually happening in the cells in those tissues that might then predispose them to becoming cancerous. So Eddie's going to talk about some approaches in this particular area later on, and I'm going to focus today on what's happening in some of the aspects of tissue characterization and tell you a story we've been developing over the past couple of years, looking at how the mammary epithelial cells might respond to increased mechanical stiffness of the surrounding stromal tissue. So one of the driving questions that we have in this area is the observation that mammographic breast density, which is a measure of the percentage of the fibroglandular tissue seen in a mammogram, is actually quite a strong risk factor for developing breast cancer, such that a woman who's got a greater than 75% mammographic density can be around 6-fold more likely of developing breast cancer than a woman with a much lower mammographic density. One of the interesting things then about mammographic density is, unlike a number of other risk factors for developing breast cancer, mammographic density is potentially modifiable for treatments like hormone-based therapies like tamoxifen can reduce mammographic density and reduce risk. So understanding the biological basis of mammographic density is quite an important thing that we want to do. Now, there's a lot of evidence in the literature that mammographic density is associated with changes in the stromal ECM, either the composition and organization, and in aspects of it being mechanically stiffer in a high mammographic dense breast compared to a low dense breast. Now, there's a lot of evidence as well over many years showing that changes in the mechanical properties of a tumor can affect tumor progressions and the proliferation and invasion of the tumor cells. But it's also quite likely then that differences in the mechanical properties in a non-diseased breast could affect the propensity of the epithelial cells to transform in the first place. And we know that there are many mechanosensitive signaling pathways in cells and epithelial cells as well, where they can sense changes in cell-cell contacts, the integrin-based adhesion complexes that interact with the extracellular matrix, and these can affect mechanosensitive transcription factors or can be directly linked to the nuclear membrane via things like the link complex. There's many ways in which cells can respond to changes in that mechanical microenvironment that may then be involved in increased risk. So what we wanted to do in this study was ask, could changes in that mechanical microenvironment surrounding mammary epithelial structures lead to changes in those epithelial cells that will put them on a potential pathway to transformation? So one of the things that's a common theme in developing 3D models to look at tissue structure is that you want to grow your cells in a matrix environment that those cells then interact with to tell those cells to behave in an appropriate way and to develop into the appropriate cells. And in doing this, the cells also modify that matrix microenvironment. So essentially, you need to create a three-dimensional scaffold that allows the cells to develop into the appropriate type of structure and organ that mimics what's happening in the tissue. Now, this works quite well with mammary epithelial cells if you grow them in a variety of different three-dimensional matrix scaffolds. So this top image is a fluorescence micrograph of a section through a mouse mammary gland, and the red stain here is the laminin basement membrane surrounding those mammary epithelial acini. And in the bottom micrograph are cells grown in a 3D culture, mammary epithelial cells, and they form a very similar type of structure. And here, stained in green, is a laminin basement membrane. So if you grow the cells in the appropriate conditions, they differentiate, they form the same kinds of structures we see in vivo, and also lay down their own basement membrane, polarizing the cells so that we have a system where these cells are essentially mimicking what we see in vivo. We can then take this model and modify various things, such as in this case, we've modified the mechanical properties. And to do this, we've used an alginate-based scaffold that's then made up with a interspersed network with extracellular matrix proteins such as matrigel, and you can then tune the mechanical properties of the alginate network by addition of calcium. So calcium cross-links the alginate, and you can tune that, and here there's AFM measurements of alginate matrigel scaffold with different concentrations of calcium that then show that we can tune and make this from a relatively soft three-dimensional scaffold to one that's much stiffer. Stiffening this scaffold then has dramatic effects on the ability of the mammary epithelial cells to form organized acini. So as you stiffen the extracellular matrix with calcium, you can get an increase in the size of the acini. They don't tend to polarize, and they, in particular, they stop differentiating. So here we have cells in a soft matrigel alginate scaffold, and they've been stained here in red for beta casein. So these ones are secreting milk proteins, and the larger acini in the stiffer alginate scaffold show much less expression of milk proteins. And you can see this if you do qPCR, they lose expression of prolactin receptor, and also they lose expression of casein 2 in this case. So the cells in the stiffer matrix are proliferating more, they're losing their polarized organization, and they're losing expression of differentiation markers. So using this kind of model system, we want to ask then, what kind of global changes are happening in these cells that might push them down a pro-oncogenic pathway? So what we did was we did global transcription profiling of cells in a soft extracellular matrix or a stiffer extracellular matrix, and we see very reproducible and specific changes in about 1,500 genes that can be either upregulated or downregulated when these cells are in different mechanical microenvironments. And in particular, one of the things we look for was differentiation markers. We see downregulation, all the things we see if we just do qPCR. So we see downregulation of all the various milk protein components and actin receptor. So we're mimicking, we can see in this global profile, all the same kind of gene changes. Now, one thing that's important when we're building these three-dimensional models or ex vivo models of any type is to get this topology right, because people use two-dimensional hydrogels to look at mechanical responses in a variety of different cancer models. And interestingly, when we grew the mammary epithelial cell on two-dimensional hydrogels that have similar mechanical properties to the ones we're mimicking in our 3D model, we see extremely different patterns of gene expression, mostly different between the 2D environment versus the 3D environment. So there's essentially, when we grow these cells on soft and stiff 2D hydrogels, we see no differences in gene expression between those, and there's far more difference between the cells between the 2D and the 3D environments. So essentially, getting the topology right in the model is extremely important for then seeing the real biological outputs.
Andrew, apologies,
So,
but if this is your 2-minute warning.
okay, sorry, yes, okay, right. I haven't got anything on here that's showing me the time. Right.
But
So,
you got me.
right. So we looked then at the kind of the pathways that are being changed here. We see lots of different changes in the things we expect, like cytoskeletal organization, actin filaments, contractility. But interesting, the biggest changes we saw were in metabolic processes and pathways, in particular things like reactive oxygen species, oxidation reduction pathways. And there's a number of papers that have come out recently showing changes in metabolic pathways in response to mechanical cues. So we hypothesized then that increased metabolic processes might lead to increased reactive products that might lead to increased genomic instability. So when we did some pathway network analysis of these metabolic pathways, in particular, we noticed that oxidoreductase activity was significantly decreased, and there were a number of aldehyde dehydrogenase isoforms that were significantly downregulated in cells in a stiff 3D microenvironment. So given that there were a number of these downregulated, we decided to look for their activity in cells. So we used a lipid peroxidation click-it fluorescent-based assay that then shows that using single-cell analysis of these cells extracted them from the 3D matrices, that there's a significant increase in aldehyde, reactive aldehydes present in cells grown in the stiff 3D matrix. So I'll go through this bit quite quickly. Essentially then, that decreased aldehyde dehydrogenase expression leads to increased genomic damage as measured by gamma H2AX staining, phospho-CHEK1 and CHEK2 staining, but also we see increased colony formation if we take these cells out of the gels and then grow them in a soft agar assay. And essentially we can rescue this either by using an aldehyde scavenger like carnosine, or expressing one of those aldehyde dehydrogenase genes under a constitutive promoter in a cell line stably, and then it essentially rescues the genomic damage and the colony formation in soft agar. So it's showing then that metabolic changes that are mechanically induced in the 3D model are then leading to increased genomic damage and potentially then making these cells make some changes down pathways for transformation. I shall go through this last bit very quickly. We've looked at some key mechanotransduction signaling pathways. So YAP-TAZ signaling is well established as a mechanosensitive transcriptional coactivator. And whereas we see YAP activation in a stiff microenvironment and it controls acinar size in the stiff matrix, it has no impact on the genomic damage in these cells. So there are classic mechanosensing pathways like YAP being activated, but it's not responsible for the changes in genomic damage that we see. Whereas one of the other mechanosensing pathways, the Rho GTPase pathway, also regulates acinar size in the stiff matrix if we express a dominant negative Rho in these cells, but it also rescues the genomic damage and the colony formation. And a dominant negative Rho also reduces the concentration of reactive aldehydes if we then measure that in these cells in the lipid peroxidation assay. So essentially what we can see then, using an assay where we can mimic the stromal extracellular matrix stiffness in a 3D model, we can then identify changes in the epithelial cells that have formed a polarized acinar structure that are changing metabolic processes leading to increased reactive aldehydes that can then lead to DNA damage. What we want to do now is increase the complexity of these models to include other cell types within that stromal extracellular matrix, such as fibroblasts and immune cells. So that's one of the areas we're in particular interested in pursuing. And then we're also using approaches to look at then what's happening in terms of the signaling pathways using proximity labeling, proteomics, and epigenetic analysis of what's happening in the nucleus. So, sorry I've gone over time a bit. It's a bit odd sat here in a room talking to a screen. The people who've done this, Amber and Haywan were two PhD students a couple of years ago who started off this project, and then Matthew and Hannah have really taken it on and finished off. And these are some of the other people who are doing various different aspects of analyzing cells in 3D models. So I shall stop there and hand over to whoever's
Sarah.
next.
Yeah, Sarah. Listen, Andrew, thank you very much. And we completely understand that this is kind of a bizarre new format. And don't worry, we're all taking it in stride. Fantastic lecture. Lots of things to think about. I just let people know, please just add your questions in the chat function, and happy to address them during the discussion. Now we'll hear from Sarah. This will be through a video, and then Sarah will be joining us for a discussion just because of a double booking.
Hello everyone, my name is Sarah Cartmell and I'm a Professor of Bioengineering at the University of Manchester. It's a pleasure to be invited to be presenting at this workshop today. I've been asked to talk to the current approaches and resources we have here at Manchester in 3D tissue models that could have potential interest in early detection research for cancer. My background is in tissue engineering with a focus on bioreactor design and 3D tissue growth in vitro. However, I'm now beginning to work with the Manchester Cancer Research Centre to translate these skills to early detection. Today, I'm going to go through some of these resources we have here at Manchester in this area, which will hopefully give a seed to foster further collaboration. So I first wanted to mention some terminology just across disciplines, because I know when I've started to work with some of the oncology researchers, and indeed when I've been here at Manchester, and indeed when I was asked to give a talk on ex vivo models, I immediately think of something else. So in terms of what I see as ex vivo, in vitro, and obviously in silico is very important, which I'll be touching on a little bit in my talk later today. Ex vivo, I always see it as a full tissue that's been taken from an organism and then that's being kept alive, and that can, so it's still valuable research, but I was a little bit confused because that's not quite what I work on. I work more on in vitro models, which is where we're taking cells and then growing 3D tissue models from that. So I don't really see that as ex vivo, and indeed, when you hear the term model, I immediately think an in vivo model rather than an in vitro model. So I've had, just to note when we start communicating in the future with research, I think that will save quite a few minutes of confusion in terms of terminology. It's worth noting. So to move on to what has already been done in the literature that most of you already will be familiar with. So we have static culture. So this is 2D versus 3D. So whilst 2D monolayer still gives very valuable information, three-dimensional cell culture systems have gained increasing interest in drug discovery and in tissue engineering due to their evident advantages in providing more physiological relevant information and more predictive data for in vivo tests. Just to have a look at these pictures that we have here. I don't know if you can see my little pointer. So this is our 2D monolayer, and then some people have been looking at a matrix for 3D culture that they grow either those single cell monolayers on top of that, or indeed 3D spheroids that are grown on a matrix such as Matrigel. Some people have been researching them when they've been encapsulated, either in individual cells or again as those 3D spheroids. Or then other researchers look at 3D spheroids grown in suspension within the media without a matrix, but again, this is still considered a 3D in vitro growth. And when we're looking at this, so again, most research regarding cancer biology is based on those two-dimensional cell cultures in vitro, as you'll be aware with. However, those 2D cultures, although they're valuable, they do have limitations, such as the disturbance of interactions between the cellular and extracellular environments, the changes in cell morphology, the polarity, method of division, for example. So these disadvantages have led to the creation of these 3D models, which are more closely able to mimic the conditions in vivo. And optimization of these culture conditions can allow potentially a better understanding of cancer biology and facilitating the study of these biomarkers and targeting therapies. So in the body, we know that cells don't really grow in a 2D environment, so conditions in a 2D system can't accurately simulate the conditions that they would encounter in vivo. So what we can see here is a schematic of the 2D systems. You can see the cells that are in contact with each other, cells in contact with the medium, and then cells in contact with a petri dish. In the spheroid culture that we saw on the last slide, we have cells either in contact with the media or with themselves, but not in contact with the petri dish. And then a lot of the work that I've been doing is using a scaffold. So you can see here a particular matrix, and this can be made out of collagen or a degradable polymer and a particular fiber size of, as you like, or indeed a hydrogel system, where the cells are actually in contact with that scaffold itself and in contact with the medium and potentially in contact with each other as well. So what can we do at Manchester to help research in cancer and in early detection? Well, we do an awful lot of biomaterials research. So if you want to have static environment but 3D in vitro sort of a tissue culture, these are just examples of some of the materials, but we have a plethora of different types of materials available, degradables, non-degradable materials. We have fibers, we have hydrogels, and again, we can have a composite of both, and we have an awful lot of additive manufacture techniques where we can actually print cells at a particular density in a particular format and build that up with these 3D scaffold constructs as well. I thought what I'd do is just show you an example here. So this is a scanning electron microscope of some work we've done within my group at the University of Manchester. In this case, we're looking at tissue engineering the annulus fibrosis for an invertible disc. But you can see that this sample here is in this G that you can see here. This is an SEM image of collagen fibers that are taken from a porcine annulus fibrosis. And then you can see that biomimicry that we can have here. So this is electrospun polycaprolactone, and the scale bar there is 20 microns. So you can see in terms of how we can create these aligned fibers with the same sort of dimensions that those cells can then adhere to and that can then influence their activity. What we've been able to do with those sheets is be able to create a multi-dimensional construct. So with the IVD, what we need is very hierarchically arranged layers. So you can see the bottom layer and the top layer in image A at the top. We're able to seed those cells onto these mats and then place these layers on top of each other and create a 3D scaffold. So you can see at day 1, day 7, those cells are viable, but also able to maintain a particular orientation, which is what we need for this particular tissue type. Just showing an example of the engineering we can do. And then we're able to use this particular device we have at the University of Manchester, this cell sheet roller. So again, we're able to then take those mats that we've created, those 3-dimensional mats, and then roll those and then create, you can see different dimensions and examples that you can see here, aorta, trachea, artery, and urethra, depending on what tissue type you're wanting to emulate. So there's potential for collaboration in this area for that static 3D tissue engineering type approach. When we're looking at more dynamic cultures, when we're wanting to actually start moving the fluid and start having gaseous exchange, and that's because you might want to control the diffusion of your waste buildup or your nutrients, such as oxygen, to the cells within your 3D construct. But you want to be able to control that. You can do that via a bioreactor. Most of you will be familiar with this type of spinner plastic approach. It's been around for many years for, just for an example of scale up of penicillin. When we're growing tissue engineered constructs, as you can see here, what we end up doing is putting the cells onto a scaffold, and we have the scaffolds that are sort of held vertically that you can see here via a wire, and we change the media probably every 3 days, probably change half that media every 3 days. And again, we have this little magnetic stirrer at the bottom, which moves around about 50 RPM at the bottom. We can have quite high throughput with data. Most of you will have these in the lab already, but again, if you don't, we can collaborate with some research in this area. We also have this setup, which is rotating bioreactor, which is a little bit different in terms of we have, these are the bioreactors, we have 50 different samples or 10 different samples in this smaller one. We purchased this commercial bioreactor from Synthicon. You can have a little look, and I'll just show you the concept. So essentially, within each of these chambers, you can inject your cells without any rotation. So this has, this whole bioreactor moves around between 15 and 25 revolutions per minute. When you're not moving it around those, you don't get any spheroid formation, you get those cells kind of aggregating at the bottom due to gravity. But when you're moving around, depending on the cell type, depends on your RPM, you can see quite quickly after 3 days, you get that spheroid formation. And the cells within that spheroid are viable all the way through, because now you're moving your culture medium rather than growing them in static culture media. The next type of 3D tissue engineering bioreactor I wanted to show you was a direct fusion bioreactor. And this is where we're physically forcing the culture medium through a cell-seeded construct. In this image we can see here, so this is a degradable, we use different scaffolds, porous scaffolds, but this is a degradable polylactic acid salt-leached scaffold that we can seed cells on. That's then placed into the bioreactor. So these are stainless steel versions. This is a polycarbonate version, but the dimensions are still the same. So we have little inlets and outlets, and essentially we seed our cells onto the scaffold, close the lid, and we have all of this sits obviously inside an incubator. We have our culture medium, and then we have our peristaltic pump, and we physically push our cells, our culture medium around and keep our cells viable within the system. So this is just a schematic of the bioreactor, the inlet and the outlet. And we're also able to obviously gather some offline data. Let me just move on for you. So have a look. So this is just some data obviously in the cell viability. So these are pores that you can see here, and these are viable and non-viable cells depending on how quickly we have our culture media flow rate on that particular scaffold. We're also able to look at different, in this case we're looking at bones, so bone-related genes, but different gene expression, different DNA, lots of offline and online monitoring we can do. And we're also able to look at in silico approaches. So this is some work we performed with Sheffield Hallam University on our particular system that we developed. Excuse me, let me just move backwards for you. Where we are able to look at where the trajectory through our scaffold. So our scaffold that our cells will be seeding on is just seen in grey here in transparency. We're assuming symmetry within the system, so we're saving computer power, so we're only imaging a quarter of that bioreactor, but you can see how that culture media is moving through, and therefore the nutrients can be diffused through our scaffold. And so we're able to design our 3D system. If you wanted a cancerous system where you wanted oxygen permeability, for example, or fluid flow permeability to one particular area of scaffold, we'll be able to control that and visualize that. And this is just to show you in terms of oxygen and carbon dioxide, not just in 2D, but we're able to do Z-stacks as well, be able to have a look all the way through our 3D system and be able to predict the concentrations of different parameters within the 3D tissue. So they were single culture approaches. We've also got co-culture approaches, and I know I won't go through, most of you will be familiar with co-culture approaches where you've got two different cell types grown within maybe a spheroid or a monolayer culture where you've grown those two cells together. These are looking at more difficult approaches that require an engineering technique to actually keep the tissue types or cells proximal to each other but not allow any mixing, if that makes sense. So one particular need is osteochondral. So again, I work a lot in orthopedics. So you can see here where we've tried to grow, the challenge is essentially having two different tissue types, so the cartilage and the bone itself, but grown a little bit integrated as happens physiologically, but again, still maintain the phenotype of the two different tissues. So just an example of how we did that was use again that direct perfusion approach where we're physically having a bone section and a cartilage section, but no mixing of the different types of culture mediums because they require different culture mediums to keep the phenotype of this cell type and indeed the tissue type in the different regions. So the culture medium, so this is the chondrogenic medium, the osteogenic medium. Again, we have our connectors that are allowing segregation of the tissue types grown together, but allow the culture of the two different tissue types next to each other. And this is just some of the data you can see. So we used a thiol-modified hyaluron and commercially available hydrogel to encapsulate our chondrocytes, in this case for the cartilage. So this was a hydrogel that we then seeded on top of this bone scaffold. In this case, we used a degradable polyglycolic acid, again a salt leach scaffold that you can see here. And again, we cultured within the system, and you can see how we can grow those two tissue types together. We were also able to look at different scaffolds within the same system. So there are lots of different osteochondral scaffolds that are being researched and being produced and innovated throughout the world. This is one at the University College London that we tested within our system, within our bioreactor. And what we've also been able to do is test the CAD, so the original design, versus the actual manufactured scaffold. So this is really important. So you have a prediction of the scaffold and the 3D tissue you're going to grow, but actually what you end up with is quite different after you've manufactured it. So this is a CT scan of, on the right hand side here, of the actual scaffold from the predicted computer design that we started off with. And again, we're then able to do some in silico modelling. In this case, we're interested in wall shear stress and flow velocity that the cells will experience as they're seeded through those different scaffolds. And you can see the difference in the profile between our predicted and our actual scaffold that was manufactured. So again, these might be some skills that would be useful to you in your research. Another co-culture approach that other people have been using, so this isn't a bioreactor we have at Manchester yet, so you can see this, the reference, but this, I just wanted to show the concept of something we're starting to work on. So this is, sorry, it's jumping around a little bit. So this is just using very simple polycarbonate membrane, which allows the diffusion of cytokines, molecules, drugs, for example, but doesn't allow the cell mixing. So it allows this systemic communication. And in this particular setup, you have cells, different cell types grown in different chambers, but they're able to communicate through this culture media. And this is something that we're starting to work on at the University of Manchester, with a collaboration with Sean Knight and Catherine Finnegan at Manchester, and also Okkan Demirci and Daniel Kim at Stanford, where we're interested in the use of this system for looking at these lung resident immune cells and what's actually circulating within the whole blood, and actually see if we can, there's any biomarkers that we could then potentially pick up from in the whole blood because of that interaction with the cancerous or the healthy tissues and what that interaction is. So we'll be able to have 1, 2, 3, multiple semi-permeable barriers within the same system. So keep those cells separate, but actually have the communication of the chemokines circling with each other. Mechanics is very important, so I wanted to show you some systems we have at the University of Manchester, recently purchased through the Henry Royce Institute. We have a lot of high throughput bioreactors, so if you do have a cell on a particular scaffold, you've already developed that system, or you want to work with us to develop that system, we're able to house those. Again, all of these are in bioreactors that we keep at 37 degrees, and again we can control the oxygen or carbon dioxide content. I'm just going to focus on this top right one a little bit. I'll show you a bit more information on how this works. So we have n equals 10 of each of these chambers. We can do relatively high throughput. The sample goes into this chamber here. So you have, and that would be a scaffold seeded with your cell types. You can have a co-culture in that system. We have compression grips. We also have tensile grips, so we can hold either something in static tension and measure online the mechanical force needed to hold that in place, or we can apply a particular mechanical regime if that is important, maybe a blood vessel that's pumping, and we can apply that regime. And we can change the culture media either on the outside and also on the inside of that scaffold if it was a vessel, for example. And of course, all of this sits inside the incubator, and we have that constantly recirculating. So that might be of interest to you. One of the last things I wanted to mention was some other research performed at the University of Manchester on organ-on a chip. So if that is of interest to you, Professor Brian Darby, I can put you in contact, works a lot in this area. And this is just showing that using his digital fabrication techniques, able to print the PDMS surface. Obviously, we can then coat with different proteins, in this case fibronectin, and then we can have a cell sheet layer sorry for the jumping around, cell sheet layer, and then another, in this case looking at kidney, so some renal cells you can see on the top, and then grow these at very high resolution. So I mentioned before Ex Vivo, so I've been asked to talk about Ex Vivo, and I just want to show you a couple of slides in Ex Vivo perfusion systems we have available at Manchester if you want to work in this area. But this is what I think of when we talk about Ex Vivo. So these are essentially some lungs, perfusion lungs. So we have access to cadaver tissue. We can do this on human tissue at low throughput or high throughput with porcine tissue, for example. So if we're looking at lung oncology, we can keep these lungs circulating and healthy, or we can look at some diseased lungs potentially, where for sort of medium terms we can keep these lungs alive for, you know, a couple of days. So there might be some early detection work that you wanted to do with this particular Ex Vivo system, and indeed we're able to perfuse whole limbs as well. So you can see the setup that James has within his laboratory, James Fowles. So you can see these porcine limbs that we're able to recirculate. Again, there's lots of different, we think outside the box on what type of testing capability we can do. We can keep these limbs being perfused through. So you have lots of different tissue types proximal to each other that you might want to test. So just to finish up, acknowledgements, lots of different funding, resources, and obviously people that have been helping create all of this data that I've shown you today, and of course yourselves, and thank you, and I'd be happy to take any questions. Thank you very much.
Great, thank you very much, Sarah, and really some exciting technologies available through a bioengineering approach. Our next two speakers are working in pancreas as well as breast cancer models, and in fact I think the two groups are actually planning on collaborating with each other. We'll start off with Dr. Claus Jorgensen from the CRUK Manchester Institute to talk about his work in pancreatic cancer. So Claus, without further ado, over to you.
Thank you, Rob. Thanks everyone for joining. Let's see if I can figure out how to share my screen here. So it always seems to be the issue. There you go. Hopefully that works. All right, so thanks again everyone for joining today. Thanks for the organizers for putting this together. So what I'll talk about today is some of the work we've forayed into over the last couple of years, essentially aiming to develop synthetic 3D models and use those to interrogate various aspects of pancreatic cancer biology. And so just very briefly, one of the problems is that pancreatic cancer, albeit being the 11th most frequent occurring cancer, is currently the 4th leading cause of cancer-related death and is projected to be the 2nd leading cause of cancer-related death by 2030. And one of the major issues is that pancreatic cancer is typically detected late. In the UK, at least, a lot of pancreatic cancers are detected when patients show up in the emergency room with jaundice, and it turns out that they have a pancreatic cancer that's already spread. And so it's actually only about 20% of patients, or even 15%, that turn up with early localized disease and are eligible for surgery. And so actually being able to move the graph and detect patients much, much earlier is probably going to be having a major impact on general survival of patients with pancreatic cancer. So this is just a very rough and tough cartoon just depicting our current view of how pancreatic cancer develops from a normal epithelial tissue through early pre-malignant stages characterized by activating mutations in KRAS and loss of the tumor suppressors CDKN2A, so that's P16 and P19. And then later on, as the disease develops into a fully adenocarcinoma, we have loss of function of P53 and SMAD4. Mind you, these are the 4 most common genetic aberrations. There's a whole host of other much less frequent occurring aberrations. But the thing that's really characteristic about pancreatic cancer is the co-evolving or de-evolving tumor microenvironment. And so there's a influx of and expansion of cells in the local microenvironment as the disease develops. So we've got infiltrating immune cells such as monocytes, macrophages, neutrophils. We got expansion of cancer-associated fibroblasts, and we got no notable increase in the number of CD8 T cells. So this is really a highly immune cold tumor and really refractory to checkpoint inhibitor treatment, at least in the current state. Similarly, there's a huge change in the biophysical environment where we have an increase in stiffness, we have an increase in the intratumoral pressure, and consequently, we have a decrease in the perfusion rate of these tumors. And so these tumors are hard to treat because it's difficult to get the drugs in, and they're also highly hypoxic. And so one of the things we've been really motivated by is, can we start thinking about developing better models to replicate some of these characteristics of the disease so we can understand how tumor cells interact with the microenvironment, both on a cellular and a biophysical manner. And so one of the big areas of motivation is that although there's a large number of really nice animal models, they are still limited in the number of genetic aberrations you can study readily. And so it's difficult to study human disease. And although organoids really have been making huge forays in recent years, they still are grown in a manner where there's a defect or there's not really a lot of cells from the microenvironment that are being depicted. So essentially we were sitting down, so this work I'm going to talk about was really run by Christopher Bello and a student in the lab in collaboration with Linda Griffith, who's a bioengineer from MIT, and Martin Humphreys, a professor for the University of Manchester, with an expertise in tumor cell extracellular interactions. And so we were sitting down and starting to think about, which aspects of the tumor biology would we ideally want to capture in an in vitro model. And so if we were to go to a synthetic model, that would be beneficial because we would be able to fully define growth conditions. And one of the current issues with Matrigel is that, about 30% of the proteins in a Matrigel prep actually differs between various batches. And so it's very difficult to actually get consistent studies when you're studying how tumor cells interact with the extracellular matrix and other components. We want to be sure that we can replicate the adhesive requirements such that the changes in the extracellular matrix are replicated in a model. We want to be sure we can replicate the biophysical changes such as stiffness changes of the microenvironment, and that we can include relevant stromal cell populations to really understand how the cells in the microenvironment affect tumor cell function. And lastly, and very importantly, we want to be sure that we can actually grow patient-derived material. And there's a lot of synthetic models out there that are really good at growing various iPSCs and murine cells, but really fail when it comes to growing human cells. And having set up a model like that, we would be really motivated to start using this to understand early disease biology and in particular the effect of the microenvironment, the impact of tumor stroma and stiffness on therapy, and then obviously translate that into human disease biology. And so the way we operate generally in the lab, but also in particular for this particular project, we tend to start with tumors and then go through a phase where we deconstruct, where we try to understand what are the individual elements of these tumors, and then we can start reconstructing it to understand how these individual parts function as a whole. And due to time restrictions, I really won't go through this in much detail, but I just really want to point out that before we got into this PEC model, we spent a significant amount of time denoting and quantifying what are the changes in the extracellular matrix as tumors develop across both murine and human tissues, such that we could have the right scaffold in terms of developing a synthetic scaffold that allows tumor cells to adhere and grow. And so the scaffold that we ended up working with is a 8-arm vinyl sulfurate that's been cross-linked with cross-linking peptides. And these cross-linking peptides have MMP sites such that proteases can act, but they also have a bacterial site such as a saltase A site that is very infrequently occurring in human and murine proteins, and therefore can be used to degrade the gel while preserving extracellular matrix proteins, other signaling cues, as well as markers on the cells, and therefore it becomes very handy when you're starting to think about studying extracellular signals and potentially looking for biomarkers in an early disease setting. These PEG gels are cross-linked with the peptides, as I mentioned, and then we include various adhesion peptides, such as this particular fibronectin mimicking peptides, a collagen mimicking peptides, and a basement membrane binder peptides, such that cells can produce their own extracellular matrix. And this becomes particularly important as we, in the earlier studies where we looked at the constituents of the extracellular matrix, identified laminin and integrin alpha-3 and alpha-6 being critical for tumor cell adhesion. And so you can essentially use these synthetic components, you can craft them together into a gel, if this actually turns out to work. There you go. And then you can embed various cell populations such as organoids. And this is essentially what it looks like from the side and from the top, where you can see these individual organoids that are growing in these synthetic gels. And this is what it looks like in the lab, where you essentially have a small membrane and you can put the gel at the bottom side and you can grow them in this media, and therefore you can end up with a very highly controlled environment where you actually grow these organoids. And so Chris have essentially very systematically gone through and identified the optimal combination of peptides to include for organoid growth. And so in this particular case, we used murine organoids, and by quantifying these individual organoids, we came up with the most optimal solution depicted over here, where we've got like 1 millimolar collagen binder, 1 millimolar basement mimetic, and 0.5 millimolar fibronectin. And we can see that in this particular case, we get a higher number of larger organoids, and we got good adhesion and growth under these conditions. So to compare what these organoids look like in the synthetic gel compared to the gold standard Matrigel, we did some cryo-EM, and we can really start seeing here how we observe formation of tight junctions in both systems. We have apical structures as well as basal structures, suggesting that the cell boundary formation and the apical basal polarity is fully recapitulated in these synthetic scaffolds. Moreover, we can grow human organoids in these synthetic scaffolds. So these are two different organoids established through local collaborations here in Manchester from surgical resected material. And we can see as the organoids are growing, we can see we have pancytokeratin as a marker for the epithelial cells. We got boundary, so intercellular boundary formation demarked by SIL1 and E-cadherin. We retained the SOX9 marker, and then we got integrin alpha-6 that we in other studies have defined as being a very important integrin in tumor cell adhesion in these particular systems. As I mentioned earlier, increased stiffness is really a hallmark of these tumors as they develop. And so you can essentially modify the scaffold by increasing the cross-linking peptides and the molar amount to generate a much more stiff gel system. And so to interrogate whether we could use this gel system to understand stiffness changes in these tumors, Chris collected a number of murine organoids and a number of murine tumors from the genetic engineered mouse models of pancreatic cancer, and as well as a number of human samples. And he then interrogated those and generated about 900 measurements for each of these samples across an area, a section of this particular tumor. And what you can see here is that there's really, you can talk about a stiffness landscape where we go from like a minor distribution in terms of stiffness, and then we got tumors that really have a broad distribution of stiffness. And we have a normal stiffness of about 1 kilopascal. The median is about 8, and the upper 95% is 26 kilopascal. And so essentially, by crafting these gels with an increased cross-linking density, we can fully recapitulate these increased stiffness landscape, going, as I mentioned, from 1 to 26 kilopascal in a very highly reproducible manner and very linear manner. And we can embed organoids and grow them in these gels. And you can see here, as measured by immunofluorescence of the mechanosensor YAP1, that as the organoids are grown in a stiffer scaffold, we can see that we engage mechanosignaling in these organoids. Lastly, we wanted to check if we could include stromal cells, as these are very critical constituents of the tumor microenvironment. And so we set up experiments where we co-cultured murine pancreatic ductal organoids together with in vitro differentiated BMDMs, so macrophages that have been treated with MCSF in culture, and then co-cultured with pancreatic fibroblasts. And each of these cells are labeled for easy visualization. So the BMDMs are orange, cell tracker green labeled. The epithelial cells are IFP labeled, and the pancreatic fibroblasts are GFP labeled. And we then co-culture them in the synthetic scaffold and then set samples away for imaging, as well as we did mass cytometry analysis. And you can see here in the imaging that we got essentially the epithelial cells and how they are cuddled and interact both with macrophages and fibroblasts in these 3D scaffolds. Moreover, using mass cytometry, we can determine that we have a preserved ratio of about 1 to 1 to 1 between the fibroblasts, the epithelial cells, and the immune cells. And more importantly, we can see that both the fibroblast immune cells acquire different phenotypes using these various markers. And so that essentially means that we have a system where we can co-culture both fibroblasts, myeloid cells, and macrophages together with the epithelial cells, and we can start interrogate how those interactions change or drive biological phenotypes. So in summary, what I hope to have convinced you is that we have a synthetic, and so this is a fully synthetic PEG model where we have highly defined growth conditions. We can replicate adhesive requirements that replicate what's observed in the tumor. We can change by physical changes such as stiffness. We can co-culture relevant stromal cell population, and we can grow patient-derived material. And these models really open up new opportunities, in particular in terms of understanding how interaction between tumor cells, a stiff microenvironment, and infiltrating stromal cells drive early disease biology. And lastly, I just really want to highlight and thank Chris for driving this project forward. As mentioned earlier, this was a collaboration between Linda Griffith lab at MIT, Martin Humphreys lab at University of Manchester, with some input from Jen Morton at CUK Beaton Institute. We had generous support from ERC and CUK to fund this project. And thanks for your attention.
Claus, thank you very much for that really exciting talk and filled with discovery elements. And again, concepts relating to stiffness and signaling, and I'm sure we'll come back to that in the discussion. It's now a pleasure to introduce our last snapshot speaker for this section of the workshop, and that's Dr. Ellen Langer. Ellen, I'll just put it over to you now.
Okay, thank you. Share my screen here. Thank you to the organizers for this opportunity to talk to you all today about the work we've been doing to generate 3D bioprinted tissue models to study both breast and pancreatic cancer. So we, as I'm sure many of you, are interested in intratumoral heterogeneity in cancer, and we think about this as being derived both by cell intrinsic differences in the genomic mutations or epigenomic alterations within the cancer cells, but also driven by tumor cell extrinsic changes like stromal cell signals, environmental stress like low oxygen or low nutrients, as well as the therapies that we treat patients with. And we know that there is a lot of crosstalk and signaling that occurs between these two compartments that ultimately ends up in heterogeneity, not only of the cancer cell phenotypes, but also of the stromal cell states. And together, we think that understanding this crosstalk can help us understand the early events in early tumor development, as well as progression of the disease. And so we are using many different models of cancer, anything from the 2D culture on plastic and co-cultures, some various 3D models, both with and without extracellular matrix, and at the far end of the spectrum, mouse models, including xenografts, patient-derived xenografts, and genetically engineered mouse models. But what I'm going to talk to you about today is are these 3D bioprinted tissues that we find really fill a gap between the simple 2D and 3D cultures that we had been doing and these mouse models. We include human cells. We can include both cultured cell lines as well as patient-derived tissues. And we use primary normal stromal cells within these tissues. And so they give us heterotypic complexity as compared to these simple cultures. And they give us flexibility and manipulability compared to our genetically engineered mouse models, because we can change very quickly what types of cells we include, or we can manipulate the expression of specific cell types to see how this influences the tumorigenic phenotypes. So this is just an image of the bioprinter that we've been using. It's an extrusion-based bioprinter system from Organovo. We have 4 printer heads that we can fill with bioink that consists of the cells that we want to print, along with a hydrogel that is gelatin and alginate based. This hydrogel gives the solution the tensile strength and rigidity to hold together in the 3-dimensional structure when we print. And shown down in this bottom left corner is our standard print, where we can surround the cancer cells on all sides with a stromal cell mix. We can print these tissues pretty quickly onto transwell membranes and then culture in standard tissue culture conditions. We cross-link the structures immediately after bioprinting to allow the tissues to hold together for 48 to 72 hours. This allows the cells to begin laying down ECM, and then we reverse those cross-links and allow the hydrogel to come out of the tissue. And so in the end, we're left with a purely cellular structure where all of the ECM that is present is laid down by the cells that we print. So it's a scaffold-free in vitro tissue model. And then on the right here is just a schematic from a paper we published last year showing the overall scheme. In general, we include fibroblasts, cancer cells, and endothelial cells within our tissues. We've done this with multiple cancer types, mostly breast and pancreas, but we've done a little bit with lung. We're able to add in different stromal cell types, and I'll talk about some of those today. We can treat with growth factors or therapeutics, and then assess with a variety of assays downstream tumorigenic phenotypes like cell proliferation and migration, ECM deposition, and then response to therapy. On this slide, I'm just showing our typical bioprinted tissue. I should mention that these tissues are small, but that they hold together like real tissue. And so we can take these tissues at endpoint and fix them in formalin, paraffin embed them, and treat them like human samples or patient or mouse samples that we would take from real tumors. We can section and stain them, or we can also dissociate and perform flow cytometry or single cell sequencing downstream analyses. Most of what I'm going to show you today is the imaging. Shown here on the top is H&E, showing our tissue-like cellular density within these structures. You can see this central cancer pocket surrounded by stroma. The middle panel is trichrome staining, showing collagen deposition. Again, all of the ECM that's present is laid down by the cells that we print. And then on the bottom and over to the right are immunofluorescent images showing cancer cells with keratin 818 staining. Vimentin in red indicates the fibroblasts, and then the endothelial cells are shown in yellow. We noted that the cells interact. Some of the cancer cells escape from that central pocket, and the cancer and stroma closely interacts. And we also noticed that the endothelial cells begin to organize within the tissues. We wanted to understand this a little bit better, so we worked with John Muschler here at OHSU to perform clarity imaging, where essentially we can remove the lipids from these tissues and then stain the whole tissue for CD31 and then do 3D imaging. And what you can see here are that the endothelial cells shown in green find each other, even though they were mixed up with all of the other cell types upon printing. They find each other and self-organized into a branched network that stretches throughout the tissue. And this is evident in the video that hopefully is playing for you on the right, where you can see these networks really stretch across the tissue. The precise role of these endothelial networks within the tissue, we haven't fully interrogated, but we know that the presence of endothelial cells and these networks contributes to the health of the tissues and allows us to culture them larger and for longer in tissue culture. As I mentioned, one of the first things that we wanted to look at was proliferation of the cancer cells in these bioprinted tissues. And so up on the left, again, is immunofluorescence with cancer cells in green and stromal cells in red. And Ki67 is stained in blue. And so these are 4 different subtypes of breast cancer, just cancer cell lines, printed among human mammary fibroblasts and human endothelial cells. And we quantified the percent of cancer cells that were Ki67 positive, and in general found that most of the tissues displayed Ki67 percentages between about 10 and 20%, with the exception of these highly aggressive Claudin low line, the MDA MB231. We confirmed this result looking in another way by infecting the cancer cells before printing with a firefly luciferase construct. And this allowed us to add D-luciferin as a substrate in the live tissues at various time points in the same tissues over time, so that we could compare relative cancer cell numbers. And once again, we see that most of the tissues increase a bit and then have kind of a steady number of cancer cells, whereas the MDA231 cells continue to proliferate within these tissues. This relatively low level of proliferation is in stark contrast to what we see with these same cells in 2D culture, where we have much higher percentages of Ki67 positivity when the cells are just plated on plastic in full growth media. We wanted to compare directly between the proliferation that we see in our bioprinted tissues and real tumor samples. And so for this, we turned to some of our pancreatic tissues, where we obtain tissues through the Oregon Pancreas Tumor Registry. And so this is the patient sample shown in the center here. With this tissue, we implanted into an immunocompromised mouse to generate a patient-derived xenograft, and that tissue is shown on the right. And then we use this patient-derived xenograft to expand our cancer cell number to have enough tissue to then bioprint, and that is shown on the left. And in all of these, we stained the cancer cells in green and Ki67 in blue, and then quantified the proliferation in all of these tissues. And we find very similar levels of proliferation across all 3 tissues. And so we think that this will be important when we do therapeutic studies, as most of many of our therapeutics target proliferating cancer cells. And so having a model system where the proliferation rate is more similar to that we see in vivo will be critical. We also wanted to look at migration of the cancer cells in these 3D bioprinted tissues. And as I mentioned, we print the cancer pocket in the center of the stromal cell mix. And so we can assess over time how these cells migrate into a stromal-rich and ECM-rich microenvironment. And so once again, we compared our 4 different subtypes of breast cancer. We had luminal, HER2 amplified, basal-like, and Claudin-low breast cancer cells that we printed into these bioprinted tissues. And as you can see pretty clearly at day 10, some of the cancer cells were very well behaved and stayed right in the center pocket, whereas others, in particular this Claudin-low line, escaped to the outside of the structure. We worked together with Youngwon Chang here at OHSU to quantify this, and he calculated a closeness centrality score by mapping the cancer cells and taking an inverse sum of their distances. And as you can see on the right, the SKBR3 showed a very high closeness centrality score consistent with their staying right in the center of that pocket, whereas the MDA231 cells had a very low closeness centrality score. We also wanted to see if we could study induced migration within these 3D bioprinted tissues. So we decided to use a very epithelial cancer cell line. Here again, these are pancreatic tissues with the HPAF2 epithelial cancer cell line, shown in green, printed among pancreatic stellate cells and human endothelial cells. And these cells in particular are, show a very tight border between their cancer pocket and the stromal cells that surround them. When we treat with TGF-beta for 5 days, however, these cells begin to migrate out into the stroma. The treatments that we perform on these tissues, however, don't only affect the cancer cells. We also noticed that the TGF-beta was affecting the pancreatic stellate cells, and we saw an increased stromal cell density in these tissues. And I point this out because I think that so often we look at the effects of ligand treatment or growth factor treatment or therapy on one particular cell type, but I think that tissues like this really give us the chance to understand how these changes in the different cell types play together to ultimately affect tumorigenic phenotypes. And so this is really where we want to focus, is how this crosstalk between the different cell types within a tumor change in response to extrinsic alterations, as well as understand if we change one particular component and knock out, for example, a factor in one cell type, how that affects interaction with the others. And so we're really using this in early tumor biology to understand how tumor cells initially reprogram a normal stromal microenvironment to contribute to initial tumor development, how different heterogeneous cancer cells can interact with the microenvironment to influence tumorigenic phenotypes, and then finally, how the environment affects therapeutic response. And so I'll give you a few examples of how we are manipulating the stroma. This is once again in our breast cancer tissues. We added in bone marrow-derived mesenchymal stem cells that are known to contribute to deposition and maturation of the collagen matrix. And indeed, when we add in the MSCs into, these are MCF7 breast cancer bioprinted tissues, we see earlier and increased overall deposition of collagen, and that's quantified, the trichrome stain is quantified in this lower left-hand corner. So again, we see increases over time in our standard conditions, but at every time point we see an increased collagen deposition when we have MSCs included in the print. And this was also seen in terms of collagen maturation in second harmonic generation imaging, where this mature collagen is shown in purple. We've added in other cell types, including adipocytes. We added these in as pre-adipocytes into the tissues and into the stroma, and this is seen, and then differentiated them in situ. And we can see this through oil red O staining of the lipid droplets in the stroma, as well as increased leptin secretion into the culture media. And then finally, in more recent work, we've been adding in PBMCs so that we can begin to interrogate immune function within these tissues. And this is work done by Isabel English, a graduate student in the lab. And so we see that these PBMCs do survive within the tissues, and she's now shown that they can be maintained in both myeloid and lymphoid lineages. So we're excited to see where we can take these models and really understand tumor and immune crosstalk. And then finally, we've been looking at therapeutic response. And so just briefly, I'll show you that indeed we see that these 3D cultures render the cells more resistant to chemotherapies. And so this was done just with breast cancer tissues. And then going forward, we're looking more specifically at the cancer cell populations by both immunofluorescence, so that we can look at the size of the cancer pocket. And these are the HPAF2 pancreatic tumor tissues treated with standard of care chemotherapy gemcitabine, and we see a response in terms of the size of that cancer pocket, and we can track this over time as well by making use of our Firefly luciferase construct, where we can measure the relative cancer cell number across different doses or across time within these tissues. So finally, I'll just put a slide in, and this is work done in collaboration with Rosie Sears and the Brendan Colson Center for Pancreatic Care at OHSU, where we're collecting primary patient tissues and isolating both the cancer cell lines as well as cancer-associated fibroblasts, and then as well over time collecting PBMCs from these patients, where we're hoping to be able to characterize different patient tumors and then begin to build back autologous tissues within the bioprinter to understand tumor stromal phenotypes, the spatial organization of these cells, as well as use these for therapeutic testing. And so with that, I'll stop, and I'd like to thank Rosie Sears, in whose lab I started this work, and we still collaborate closely on many of these projects. Isabel English and Zineb Doha are two graduate students who are doing a lot of the bioprinting work, along with Megan Turnage, a fantastic research assistant within the lab. And Brittany Allen Peterson started this project with me. She's now at Purdue University, and Nick Ansersky was the technician who really started this with us. I'd like to thank others at OHSU who've helped analyze and assess these tissues, as well as the group at Organovo who really was there in the beginning and got us started with these tissues. And so with that, I'll stop, and I'm happy to also take any questions.
Ellie, thank you so much for that. And if you could turn on your video, and I'm going to ask Andrew, Sarah, and Klaus also to turn on your video, because we have a bit of a panel discussion, and all of the presentations were highly complementary, slightly different, but also bring up some similar questions. So I want to just focus on, if I might, and then we have some specific questions within the chat function, but perhaps just having a general discussion around some of the elements, and that is, I want to come to the immune cell again, and just what do we think are absolute for the people who are on the call right now, absolutely the types of immune cell interactions that we feel confident that we can address with these ex vivo systems, and what do we think we absolutely cannot address with these systems? Because you know, if we took at face value everything that was said in the last 4 talks, my God, everybody would be changing ex vivo systems because there's nothing you can't do. Technology is cool, and Sarah has really interesting instruments, right? But I do think there probably are a few limitations as well as the opportunity. So maybe I'll ask Klaus to start off with, and happy for others to chime in. But Klaus, just in terms of you thinking about the full recapitulation of the tumor microenvironment, what are your thoughts around the immunophenotyping of primary tumors versus what we can achieve ex vivo?
I think that's a very good question, Rob. I can only speculate. I mean, this is something we've just started looking into at the moment, and we started using like the bone marrow derived macrophages that were differentiated in vitro, because that's the kind of cell type that we could best get access to and start incorporating. And some of the phenotypes we are seeing, at least using the mass cytometry, replicates what we would see in the tumors in vivo. So looking at immunophenotypes in vivo. Beyond that, I mean, including immune cell populations that require, you know, going to the local lymph node for antigen cross-presentation, that's obviously going to be much more difficult, and it's going to require a different setup in terms of microfluidics, more like organ-on-chip systems. And how far we're away from that is a good question.
Fair enough. Ellen, what do you think? You got us into this a little bit at the end of your presentation, so.
Yeah, I'll agree. I think it is early days with our system. We're excited to see the cells survive, and I think that one of the most direct approaches that we're interested in is how different cancer cells, depending on their oncogene expression or their phenotype, differentially program that immune microenvironment toward tumor promoting versus tumor inhibiting. And so I think those kinds of things are most approachable today, but I do think that pushing the limits and seeing how far we can take the system, maybe connecting with organ on a chip kind of technologies, will be interesting to see what's possible.
Great. Andrew, did you want to add anything, or?
Nothing specific, but I think there's clearly in the normal tissue, there are potential links between immune and inflammatory cell type changes that are associated with risk that might then either be driven by the stromal changes or be driving the stromal changes. So thinking of how to incorporate those into the models is important, but I think, yes, it is early days, and maybe even more so than with normal tissue rather than incorporating the cancer tissue.
Yeah, Claus.
Just one more comment. I kind of lost my train of thought there for a
Yeah,
second.
no, okay, we had an editorial comment from a young one in your virtual lab.
Yeah, exactly. I do think one thing where these models potentially could be interesting in terms of further interrogation is to understand some of the cues that attract various immune cell populations. Because if you got the tumor cells and other stromal cell populations together in a gel, and you can then include your immune cell populations and actually see how they are attracted and the resulting immune phenotypes that are coming in there, I think that's a potential advantage of those models.
Right. Sarah, I'm just going to pick up a little bit because we have a question. I think actually it comes a little bit from Claus to Ellen, but it's something that I think we need to address, and that is the importance, but also modeling of endothelium and blood flow within the cultures. And I think you have a question, Claus, to Ellen around parasites, but just in terms of blood flow, I think you have described, Sarah, some models that you have. I guess the question to all of you is, should all of your cultures have endothelium? Should they all have that as a critical component in terms of the microenvironment? And do we have any evidence that the amount of, I guess, endothelium changes the biology? Because I thought Rosalie showed it in Portland a couple years ago as an early detection that she was describing some effects of endothelium, but it might have been a different one, a different person, Ellen. But it certainly came up when we were discussing about, admixtures of endothelium or not, and this could potentially have profound, effects. So maybe a little bit on some of the technology, Sarah, and then kind of open to the other three individuals about your thoughts on this.
Yeah, it's a very interesting question. So when we're looking at tissue engineering aspects, and we've been looking at co-cultures, very similar sort of studies, that very nice studies that you showed, Ellen, similar sort of results where we're seeing the spontaneous coming together, those endothelial cells and the blood vessel formation for tissue engineering, which is really exciting. We were using the direct perfusion systems more because we wanted to be able to do the same job as the blood vessel system rather than biomimic the blood vessels, right? So we wanted to be able to transport particular nutrients such as oxygen, just to keep our cells viable. That was what we wanted our system. And I wonder whether or not, because when I was looking at some of Ellen's work, and I didn't know whether or not it was the presence of, it's probably multifactorial, but presence of the cells and the communication to the cells and the signaling molecules coming from those, but also that oxygen availability. That would maybe make a change, and particularly some of the systems in terms of not just with endothelial cells, but also when I think Claus was showing the cross-linking, and whether or not that would affect the oxygen permeability within that system as well, and whether or not the cells would, some of the results you might see might be because of the oxygen tension changes rather than the mechanical stiffness. But in terms of the technology, what we were saying and what we can do, we do a lot of a lot of CT methodology development. So we have a very high resolution computed tomography, and we're able to sort of track fluid movement through different structures. So in our tissue engineering systems, we're looking at tracking using iodine contrast agents, tracking through porous systems. But actually we could, if you wanted a functional endothelial vessel, we could actually see if there's any transport mechanisms through that as well. That might be quite interesting to look at.
Yeah, absolutely. Ellen or Claus or Andrew, do you want to speak a little bit to kind of blood flow and endothelium? Ellen, I'll start with you.
Yeah, so we see the endothelial networks form driven only by the cells that we're putting in, that they find each other and self-organize, which we find fascinating and we find is necessary to be able to build bigger tissues. But we haven't pushed the biology of that very much at all. And so whether or not those are helping conduct nutrients into the core of the tissue, we haven't tracked, but we need to. And then also whether or not they're preventing a hypoxic core, we haven't looked into, but we know that it's on the list of kind of determining what that function of those cells is. Does it allow for cell survival because it's actually transporting something, or is it simply signals from those endothelial cells, paracrine signals that are helping support survival? And we haven't pushed that angle yet.
And of course, for chemosensitivity testing, it might also be important in terms of passive diffusion versus coming through the vasculature. And you know, as someone who works on hypoxia, we know it's actually very important. Claus or Andrew, any comments about it?
But so we haven't started to include the endothelial cells. It's certainly on the to-do list. I think it would be very interesting to do. We can see, you know, if we grow the cells in slightly larger gels, the core, at least the cells in the core of the gel, they do not look very happy and they start dying, and that's probably, we suspect, is due to hypoxia. So we reduce the size of the discs for now. But I do think, you know, perhaps including some endothelial cells in the future would definitely be interesting.
And maybe there are a couple of questions, you know, just on the chat about normal tissues again, and kind of comes a little bit to Andrew, but also to the pre-malignant tissues that may have oncogene and activation, but not, you know, and potentially immortalization, but actually don't have transformation. And I guess, does anyone here that are, you know, the experts who are part of this panel see any problem with actually trying to grow any normal tissue ex vivo, any normal tissue at risk for early detection studies in terms of modeling potential changes and acquisition of genetic instability and kind of tracking these things in the pre-malignant but high risk tissue?
We've managed to grow primary normal mammary epithelial cells in 3D cultures for 3 to 4 weeks. So you can potentially, that's actually just on just the epithelial cells. So it will be interesting then to see how those are different when we incorporate other normal cells into a surrounding stroma. But I think there's certainly possibilities depending on the, I know most of what people are doing here is with tumor cells, but from then the early detection perspective, we want to be looking at normal cells.
Yeah, or macroscopically normal cells.
Yes, or cells isolated from a higher risk tissue.
Precisely.
They
yeah.
haven't, yeah.
Yeah, and that have field defects, et cetera. And I think that's really an exciting era. But you know, I would certainly propose, you know, based on all of your work, that you know you need to get the systems going in one aspect to actually drive it through much more complex systems, which, although it's simpler in a cell model, it might be much more difficult in terms of the growth activity, the growth factors that are required, et cetera, you know, across different tissues. Just some specific questions just to end before we move on to our trainee presentations. Joe Parsons had a question kind of for everybody in this, but it was specific, Klaus, that in breast cancer, some surface markers can be lost when cells are grown ex vivo. And I guess the question was, do you have any evidence that any surface markers or stem markers are actually lost when you take cells out of patients and put them into the ex vivo models? And Helen, feel free to comment on it as well.
So the markers we've tested so far, they seem to be preserved. So technically, these gels have a different background when we do IF and IHC. And so actually doing your box standard where you take your gel and then either include an antibody and stain by IF is much more difficult because of the structure of these gels. So we have to do a section and first and then stain, and we still get background. But depending on the methodology, we've been able to detect most of the cell surface markers we've looked at so far. And we've done a more comprehensive study in the KPC tumors, and we're going to start comparing that across to see, you know, whether they're preserved similarly. Joe had a question also regarding sortase, and that was one of the reasons why we started using this system, because you can kind of get the cells out with the sortase, that's a bacterial enzyme that really have very low specificity or doesn't recognize most mammalian receptors. And so that means that you can better preserve cell surface receptors, which could also be a technical issue in other systems.
Fair enough. And we have one more question that I'm going to ask you, then we're going to move to the trainees just because of the timing at 5:31. And this was actually a question to Ellen. And this was a very simple kind of technical question, but do you have any information if the bioprinting process itself puts cells under stress? Does the extrusion through the probe change at all mechanical forces and signaling?
Yeah, so we haven't tested that directly. And there is some shear stress on the cells, but the viability of the cells seems to be very high. So we have 250 micron needle. So it's not, we're not trying to send the cells through a tiny gauge in order to print. We're putting them through a 250 gauge needle. And in general, the cells are pretty viable coming through that. We haven't tested signals of mechanical stress specifically, and we should probably do that.
And I guess, just to end this panel and move on to the other trainee lectures, one of the queries, what's the control? What's truth, right? And the reality is that signaling, within the solid tumor versus signaling ex vivo, we're trying to mimic a number of things, but at the same time, we get to change, microenvironmental parameters. And I just think that these are separate in ex vivo systems that we allow us to really control a number of variables and may never completely mimic the human situation. But really, I don't think anyone would argue that you would do one or the other in the sense that both of them are probably highly complementary, right? So we're going to move on then to two more presentations, and I'll ask Adi Skaria to basically load up his presentation, and he is a postdoctoral fellow in Andrew Gilmore's laboratory, and we'll be talking about hydrogel scaffolds. And then this will be followed by Helen Clough, a PhD candidate in Sarah Cartmell's laboratory. So Adi, take it away.
Thank you. Just going to share my screen.
And just as you're doing that, there are a number of questions we didn't quite get around to in the panel that people are asking in chat. We will actually collate these questions and on your behalf, email them to the panel members so that we can get your questions answered. Adi, over to you. Thank you.
Hello, thank you for inviting me to give this talk. As Rob said, I'm a postdoc working with Andrew Gilmore, Josh Swift, and Marco Domingos, and right through there, there's expertise within cancer research, biomechanics, and engineering. So the main focus of our group is to develop biomimetic hydrogel scaffolds for breast cancer research. And today, this talk, I'm hoping to give a little bit of insight about how we're trying to approach this scenario. So our aim is to leverage our understanding and advances in biology to develop biomimetic scaffolds. And in case of breast cancer, which is a very complex microenvironment, we know that normal mammary ECM is significantly changed. There is an increased production of fibrous ECM promoted by activated fibroblasts in cancer cells. This leads to increased deposition of collagen, for example, and collagen cross-linking by increased LOX enzyme activity. This, together with the secretion of other basement membrane proteins, such as that of fibronectin, for example, leads to higher stiffnesses, which is the hallmark of the condition in terms of breast cancer, as associated with high mammographic density, which is the hallmark of the disease. So we are currently undertaking a lot of work in our lab to characterize these tissue microenvironments into really to understand what is different from normal to breast cancer tissues, to get a very detailed overview of spatially defined characteristics. Where I am placed in our group is understanding the biomechanics of the tissue. So this is the other side of the puzzle, essentially. It's understanding biomechanics, which is where my work is placed. So from biomechanobiological studies, we can now appreciate that biomechanical properties of a matrix play a central role in shaping tissues and maintaining homeostasis, as well as that it is perturbed in many cancers. As with breast cancer, and using collagen as an example, where this increased deposition, cross-linking, and alignment are all central features of the disease. My aim is to understand or use a range of design parameters to implement these tissues to align native tissue mechanics to that of a synthetic scaffold. For example, by implementing features such as the fibrous architecture with the right bioadhesive cue, controlling cross-link intensity to control pore sizes, degradability to allow remodeling of the matrix, and so on and so forth, and an overall control over mechanics. And this is what I'm hoping to aim with developing more synthetic biomimetic scaffolds. So I'm essentially starting from bottom up, essentially. So as a first step towards synthesizing any scaffold is to understand what the environment is made of. And we are all aware that within a basement membrane, there's two independent polymeric networks, one of laminin and one of collagen 4. And this forms the main architecture of the basement membrane, whose mechanics and function is driven by the assembly of this essential basement membrane. And what I want to focus on is laminin, because laminin is the foundational structure of protein that initiates basement membrane organization and assembly. And these two networks are essentially linked together by molecules such as agrin and perlecan to hold the structure together. So what researchers struggled when it comes to incorporating laminin into those matrices is that laminin, when we buy this particular molecule, and it is highly heterogeneous, and there's a high cost associated with it. When people have tried to incorporate laminin into non-covalently into a matrix, there's an usual problem associated with diffusion of it out of the matrix. And when researchers have tried to covalently functionalize a matrix with this bioadditive cube, there's difficulty in controlling its orientation, selectivity, and it permits limited exchangeability. Laminin is a molecule that has been investigated over the past 40 years, but only recently, for the reasons that the main reason why they have been investigated is to identify specific sequences within that, or cryptic sites that are essential for tissue engineering or for the purposes of downstream kinds of targets. But from this, what has been identified is a minimum integrin binding fragment of the laminin molecule, which conserves all of the integrin binding regions, integrin binding globular domains, and a short fragment of the triple helical coil structure. So in our lab, we're hoping to take advantage of these new information and really start to think about how to selectively modify these laminin molecules to produce N-terminal modifications and incorporate that into a matrix. Think about how to make a dynamic system, how to engineer high affinity interactions to other basement membrane proteins, such as that of agrin, for example. And what this allows us to do is then to use this laminin as a foundational molecule within our matrix without having any limitations of usage, and with high specificity and high selectivity. So by hoping and by really establishing a cell line that secretes these proteins, we are tethering these to biomaterials. We're hoping to produce a biomaterial scaffold with a range of desirable scaffold properties. For example, we're interested, as previously mentioned, we're interested in a fibrous matrix. We are interested in a capacity to remodel, especially that of self-healing properties, for example, because then we can take advantage of technologies such as 3D printing, which is quite established in Manchester, where this image shows a gel-based printing system where gels are printed into a sacrificial gel and then reticulated afterwards. And through this way, we can, by adopting this kind of technology, we can start to think about producing a high throughput system where we can have cells, as Helen showed previously, with epithelial components and stromal components in a spatially orientated manner. And this is what I'm hoping to do with my post-structural position, which is basically to map and tissues or produce a multi-layered structure with integrated mechanics in terms of stromal, epithelial, and adipose tissue components, and really build a 3D system that's truly biomimetic. And I hope we can produce these systems in the long run and really begin to understand how tumor initiation occurs and how and we can really start to think about how to decouple the mechanics from molecular events and really understand the mechanobiology of these processes as well. Thank you for listening. Happy to take any questions.
Great, thank you very much, Adi. Are there any questions for Adi, just in terms of, we'll take one question and then we'll move on to another? And again, feel free to email the organizers. We can always share your questions with the speakers afterwards. Okay, Addie, thank you very much. Again, a great expose of the technologies and approaches and rationale with the lab in Manchester. And now, Helen, I will invite you to give a short presentation on your work, please.
Thank you. Just let me. Okay, so my name's Helen and I'm a final year PhD student based in the biomaterials group here at Manchester. And today I'll be talking a bit about my project, which is using self-assembling peptide hydrogels to mimic the tumor microenvironment in vitro. So self-assembling peptide hydrogels are made from short peptide sequences, which assemble first into beta sheets, then into nanofibers to form a self-supporting gel. So these hydrogels are synthetic by design and are chemically defined, and this offers a significant advantage over naturally derived hydrogels. For instance, if we look at the fiber diameter of major gel and collagen, popular biomaterials used in cancer research, and peptide alpha-1, the peptide gel I'm interested in, you can see that the fibers of peptide alpha-1 are much more uniform in size and significantly thinner than that of collagen major gel. And looking from a stiffness point of view, alpha-1 is also considerably stiffer than that of collagen major gel, showing that it can mimic the mechanical properties of tumor tissue more faithfully. So the aim of my model is to try and mimic vascularized breast tumor in vitro using these peptide gels. So I'm using two different cancer cell lines, MCF7s representing early stage cancer, and MDA and B231s representing metastatic breast cancer. And I'm also using HUVECs and MSDs to create blood vessel-like structures within the constructs. So the point of this model is to try and aid our understanding of tumor biology and progression by observing cell-to-cell and cell matrix interactions, as well as providing a novel platform to test chemotherapeutic drugs. So we use live dead staining and PK green analysis to show that both cell lines are very much happy within the gels over 14 days, showing that the peptide gel can support the growth. We use histological staining to show how the cells behave within the gel. So H&E staining of the MCF7s shows that the cells tend to aggregate and form spheroids within the gel over 14 days. And Ki-67 staining also shows the cells are proliferating and depositing large amounts of VCM. MDAs, on the other hand, behave much differently. They tend to remain dispersed with loose aggregates seen by day 14. Although we do, so they do proliferate, although not much collagen-1 deposition observed. So as mentioned earlier, hypoxia is a really important feature of solid tumours. As tumours outgrow their oxygen supply, this results in hypoxia recurring in the core, causing cascade of events resulting in angiogenesis. So we wanted to see is hypoxia occurring in our model. So the images you're looking at are the whole gels. And so you can see that over time, the number of hypoxic cells does increase over 14 days, showing that our model can mimic this feature. We assess the invasive potential of both cells by embedding cell-laden peptide gels into the collagen and major gel. So you can see over a 21-day period that the MCF7 cells will degrade the peptide matrix and invade either major gel or collagen, as you can see by the process of cells, although we show that cell invasion and metastasis can be mimicked more closely. So we use TEM imaging to look at cell material interactions. So we see that the MCF7 cells actually internalize peptide alpha-1, as you can see by the pockets here. Although they don't do that with collagen, instead we see more anchoring of the material. Again, MDA and B231 cells don't internalize the gel, they just tend to explore the material with processes, and we don't see this with collagen. So looking at the MCF7s and peptide gels more closely, firstly you can see how enormous these spheroids become, but also you can see in more detail the enveloped gel within the cell bodies. And this, to us anyway, highlights a novel method of introducing drugs to cancer cells in vitro. So finally, I just want to touch on some work we're doing with a group in Sweden, which is looking at how we can spatially determine gene expression of EMT markers, so providing histological context to traditional gene expression. So the current data shows that the peptide gel doesn't interfere with methodology and that genes of interest are detected. So in conclusion, Alpha-1 is a suitable platform to model cancer in terms of its characterization, and can also support the growth and viability of 2 breast cancer cell lines. We see features such as hypoxia, invasion mimics, and the MCF7 cells appear to be internalizing peptide fibers, which highlights a novel opportunity for drug delivery and future work. We'll look at EMT and drug efficacy. And with that, thank you for listening.
Great, thank you very much, Helen. Fascinating talk as well. Just can you describe a little bit more for me the work that you're doing with Sweden in terms of a little bit more granularity?
Yeah, sure. Obviously, due to time constraints, I didn't go into it too detail. But basically, we're using sections of cell laden gels, and they're using they describe it as a lock-and padlock approach. So basically, they can detect the gene products within the sample. So at the moment, we've only been looking at seeing are the samples actually appropriate using the methodology. So we haven't gone too much into the nitty-gritty of the EMT markers themselves. So at the moment, it's just very preliminary work.
No, fair enough. Thank you very much. And John Cheng has a question, which is interesting that MCS7 is more invasive than MDA231. Can you comment on that, please?
Yeah, so our current theory is that the peptide gels can't really support the aggressive phenotype of the MDA MB231s, whereas the MCF7 cells are far more active within the gel. I guess a reason for the lack of invasiveness from the MDAs could be there's no like adhesion cues in the gel, maybe, so there's nothing for them to adhere to and therefore elongate. Yeah, it's something we're trying to investigate.
Thank you. And Adam Pickard asked a question, is the EM showing internalization of fibers or cells wrapping around the fibers?
That's a really good question. So we do see that the cells do tend to wrap around the gels, but also we do see that really like in the center of the cells enveloped gel. So we think maybe that as the cells are forming spheroids, that they're trapping gels, and also that they're enveloping gels into these vesicles.
Great, thank you very much. Thank you very much, Helen. I want to really draw the workshop to a close now and actually thank all of the speakers today. Again, I think we really brought out some fascinating opportunities to delve into early detection across a number of technologies that are available within the Alliance for Center for Early Detection, and then of course also intramural programs within the University of Manchester. The Manchester Cancer Research Centre is very supportive of build and ex vivo models, and so for those of you who are on the virtual line, quote unquote, that have interest in this, please stay tuned, because as COVID-19 winds down and laboratory activity starts to wind back up again, we're really going to start to explore how we can, again, develop models across different normal tissue histopathologies and also across different types of the tumor spectra, and really answer questions not only with respect to early detection and tumor progression, but also, as is pointed out, novel models potentially for the metastatic niche, and that includes both thinking about lymph nodes, but also about bone metastasis. So with that, I'd like to thank all of the speakers today. Just to remind everybody that this lecture will be available through the MCRC website as well as the ACE program, and it'll be on YouTube after we edit it for a YouTube video. This will be one of the first of, we hope, 4 or 5 workshops in early detection and about 10 workshops from MCRC across a number of different questions that are burning within cancer biology. And I want to thank the team that organized this, Joe Clark, Georgina Binney, Martin Bone, and the ACE crew for today's session. So with that, I'll leave you to enjoy your days, and we look forward to another great session again, with a combination of University of Manchester as well as ACE investigators. And we certainly hope coming out of this session, there'll be a number of applications to the ACE scheme that really looks at early detection and the problem of early detection using these models. So thank you and enjoy your day.